Hello friends, welcome back to my YouTube channel, The Microbiology Digest. So today we'll be discussing on a different topic, that is growth and reproduction of bacteria. So when we say growth, there are some principles in case of bacteria. So growth, which is in general, it denotes the orderly increase of all constituents in a cell. So when we apply this term growth to bacteria, it generally refers to the changes in the total population rather than increase in their size. So here what happens in case of bacteria when we say it is growth? So here it indicates the changes in the total population, not in the changes or size changes in the size of individual bacterium. So when there is a balance between these two processes, the net result in case of bacterial growth is the increase in the total mass, or means total population. There will be, when we say it is growth in case of bacteria, so there will be increase in the total population of the bacterium. So when we want to conduct any studies in microbiology or bacteriology, so we need to cultivate the organisms in the laboratory. So for which it we should be well acquainted with the principles of this growth of bacteria, which will aid us or which will help us in proper cultivation of the bacteria in the laboratory. Now, how bacterial cell divides? So there are different methods, but principally there is only one method, that is binary fission, through which almost all the organisms they divide. It is the most common mode, and the actual process which is called as transverse binary fission. Okay, from one cell, so there will be one cell will divide into two different or two new cells. What happens? One single cell divides after developing a transverse septum or cross wall in the cell. So ultimately, this septum will lead to development of or uh, growth of two individual cells from a single bacterium. And this process is very rapid, and it helps bacteria to grow very fast in favorable conditions. So the method by which bacterial cell divides, it is the most common one is the binary fission. There are some other methods also. Some bacteria, we may see different methods by which they divide. One is budding, then third one is the fragmentation, and fourth one is the formation of sporangiosperms or conidiospores. So although these three methods are there, but they are very less common in comparison to the first one, that is binary fission. So binary fission is the most common method by which bacterial cells divides. Now, basic requirements for our topic of discussion, today's discussion is on bacterial growth. So for bacteria, or to get optimum bacterial growth, so it there are some factors which acts on, or which aids or which helps on bacterial growth. So first type of factor is the nutritional factor, then second type is the physical factor. So two factors, nutritional factors and physical factors. So nutritional factors, in our earlier discussion already we have discussed in detail. So link I have provided, you can go through, or you can see if you have missed. So then what are other physical factors apart from nutritional factors? So there are some physical factors are also involved, or which also have some effect on bacterial growth. So physical factors includes the temperature, oxygen concentration, pH, etc. So these factors, physical factors, if we are well aware about these factors, so it will help us in successful cultivation of the bacteria in the laboratory. Now, physical conditions one by one we will be discussing. First physical condition required is the temperature. So, temperature for all bacteria, there is a temperature in which bacteria favors to grow. So, that temperature is called as optimal growth temperature. So, it is different from bacteria to bacteria, but this is the temperature. Optimum growth temperature is the temperature which will allow rapid growth of bacteria within a very short period of time. So, that temperature is called as optimal growth temperature. And on this basis, as I said, required temperature requirement is different from bacteria to bacteria. So, we can divide bacteria into three different groups based on the temperature requirement. First group is called as psychrophils. Psychrophils, they are able to grow at 0 degree or lower, but their optimum temperature, it is 15 degrees centigrade or maximum is 20 degrees centigrade. So within this range, they favors to grow, that is their optimal growth temperature. And there is another term which is called as psychrotrophs, or they are also called as facultative psychrophiles. So this is a different group of bacteria within psychrophile group. This group of this bacteria which are called as psychrotrophs or facultative psychrophiles. So these are the organisms that are able to grow at 0 degree, but they will grow best at a temperature range of 20 to 30 degrees centigrade. Means although they are included under the group psychrophiles, so their best grow temperature or their optimum grow temperature is 20 degree to 30 degree. They can grow at 0 degree, there is no doubt about that, but their optimum grow temperature is 20 to 30 degrees centigrade. Then second group of organisms based on the temperature requirement is mesophiles or mesophils. So their optimum temperature is from 25 degrees centigrade to 40 degrees centigrade. So within this range, this group of organisms, they can grow. Then third group is the thermophiles. So their temperature, from the name or term itself, it indicates that they grow in a higher temperature. So optimum temperature is always above 45 degrees centigrade. So that is why they are called as thermophiles. Okay. Second physical condition which is required for bacterial growth is the pH or hydrogen ion concentration. So like temperature, pH is also an essential requirement for optimum bacterial growth. So optimal range is 6.5 to 7.5. Within this range, pH range, bacteria prefers to grow and multiply. So based on the pH also, we can divide the bacteria into three different types. Neutrophiles, so they grow based at this temperature, which is favorable for almost most of the bacteria, 6.5 to 7.5. So majority of the bacteria, they are neutrophiles. Then the second group based on pH requirement is acidophiles. They prefer to grow based in acidic conditions or lower pH. Then third group is alkalinophiles. So they grow based at alkaline conditions. Then third requirement, physical requirement, is the oxygen. Oxygen is one of the principal requirement. So bacteria, they vary in their requirement of oxygen, and based on this requirement, there may be four different group of bacteria based on the oxygen requirement. So first group is the aerobic bacteria. So this group of bacteria, they require oxygen for growth, and they can grow when incubated at an atmospheric air, where there is 21% of oxygen must be there. Means they require oxygen for growth and multiplication. So this type of organism or bacteria, they are called as aerobic bacteria. Then second type is the anaerobic bacteria. So they cannot grow or they even die in presence of oxygen. When we incubate these type of organisms in presence of oxygen, they cannot grow or sometimes they even die. So they are called as anaerobic bacteria. And in this case, anaerobic bacteria, oxygen is generally toxic and they cannot grow in presence of oxygen. Then third group based on oxygen requirement is the facultative anaerobes. So they can grow with or without oxygen. Okay, generally they do not require oxygen for growth, but these group of organisms, they can use oxygen for their other processes like energy production. Okay, they are not inhibited by, although they are anaerobes, so their growth is not inhibited by oxygen. Means they can grow both in presence of oxygen or in absence of oxygen. Okay, but growth is always better when oxygen is present. They are called as facultative anaerobes. Then last group is the microaerophilic bacteria. So this group of organisms, they require a very low amount of oxygen. Okay, low level of oxygen is required for growth and multiplication of this group of organisms. They are called as microaerophilic organisms. Now, bacteria, when bacteria are inoculated to a new nutrient medium for during the cultivation process, so the stages are different stages of bacterial growth. So that can be divided, or for understanding we can divide the different stages of bacterial growth into 4 different phases. So when we inoculate an organism to a fresh medium, so bacteria starts multiplying if we incubate them at appropriate temperature, and this growth of organisms, upon incubation, bacteria will start multiplying, or there will be growth of the bacteria. So when this growth of the bacteria is plotted against the time, so a specific curve is generated. That curve is called as bacterial growth curve. So typically the bacterial growth curve, it contains 4 different phases. First is the lag phase, second is the log phase, third is the stationary phase, and fourth is the phase of decline. Now, when we are plotting, there is a term called generation time. Generation time is often we need to calculate when we cultivate the organisms in the laboratory. So this is generation time is the length of time required for single bacterial cell to yield 2 daughter cell. Means when a population, this is the time required by a bacteria to make its population completely double to its original population. Okay, this time is called as generation time. So generation time average in case of bacteria, we have seen it ranges from 1 to 3 hours, but there are some organisms or bacteria where this generation time is very less. In case of, suppose for example, Escherichia coli, the generation time is hardly 20 minutes. So during this 20 minutes, the parent population, it completely gets doubled during these 20 minutes. So that is the generation time. Now this is a typical bacterial growth curve. First phase is the lag phase, second phase is the log phase, or it is also otherwise called as exponential phase, third is the stationary phase, and fourth is the death phase or phase of decline. Now we will discuss one by one. First phase, lag phase. So when we inoculate the organisms to a new environment or new medium, so the organisms or the cells, bacterial cells, they need to adapt themselves to the new environment. So during this period, the population, it generally remains unchanged, means there will be no growth of the organism, but still the bacterial cells, they are trying to adapt to the new environment. Okay, apparently there will be no uh, growth of bacteria, no individual cells will increase in size, but they are physiologically very active. Okay, and they are synthesizing the new protoplasm. Okay, so this phase, this is also called as adaptation phase of the bacterial cells to the new environment. So this is the lag phase or first phase of the bacterial growth curve. Next, the second phase, it is the log phase or the logarithmic or exponential phase. So in this phase, what happens? There will be rapid growth of the organisms during this phase. So this phase, actually, this it starts with rapid multiplication of the organisms, and there will be sharp increase in the bacterial population. So maximum cell division occurs at this phase, and the population, it gets completely double, and all the cells, they will be nearly uniform in their size, because new cells are continuously forming during this phase. So this phase is called as the log phase or exponential growth phase, and as the cells of in this phase of bacterial growth are uniform, so most of the studies involving microbial cultures or microbial other studies, so cultures from log phases are preferred because cells remains uniform during this phase of growth. Next, the third phase is the stationary phase. So after uh, exponential growth, so here in this phase there will be the cell division, or there will be no cell division will take place here. And why this happens? Because there will be depletion of the nutrients, because whatever nutrients are present in the medium, so they all the nutrients have already been utilized, and there will be accumulation of the metabolic products, or which are waste product of the bacterial growth. Because of this, the bacterial cell division will stop and the cells will start dying. Okay, so this is the stationary phase, and the graph, it will maintain a steady line. That is why it is called the stationary phase, and the number of cells produced here in this phase, it is equal to the number of cells are dying. Okay, so this is both equal, the number of cells are produced and equal number of cells are dying. So that is why it maintains a stationary line. So this stage is called as stationary phase. And the final phase of bacterial growth curve is the phase of decline. So decline phase, it is the as the name says, it is the phase of death. So in this stage phase, the bacteria starts dying, and the number of bacterial cells or dying bacteria, it is more than the number of cells produced. Of course, there will be no more cells are being produced, only uh, death of the bacterium will take place in this phase, because no nutrients will be available, and the accumulation of waste products from the previous growths, so this will act on there will be harmful effect of these waste products on the bacterial cell, and because of these reasons, the number of viable cells in this phase, it declines sharply. Okay, so rapidly there will be uh, number of bacterial cell, or growth of the cells will be falling down, and more number of cells will start dying in a very faster rate. So this phase is called as phase of decline. These are the four different phases of bacterial growth curve. So this is what today I was supposed to discuss in a very brief manner. So I hope this is will be helpful. So if you find, please like, share, and comment this my channel. And till then, I'll again in our next discussions, I'll come again with a new topic. So till then, bye bye. Thank you.